thank you, Professor Wesig, for this introduction, and I'd like to thank the organizers for the opportunity to present our work here. I enjoyed very much the introduction this morning by Professor Wesig, which made my job easier, and I also wanted to comment and to assure you that you're not swimming alone against the stream on the issue of what are nano drugs, both the definition and the other aspects.
Thank you very much.
So I'd like to present to you a somewhat different aspect of drug delivery, which is the computational part or theoretical approach to drug delivery, which is not focused only on drug delivery because the algorithm that we developed is a very generic one and is not even only restricted to drugs but also to other complex issues in other fields such as transportations and communications and financial applications and so forth. Anyway, my lab is focused on this issue of drug discovery and drug development by computational methods. And the work that I'm going to describe here is a collaboration with the lab of Professor Barnholtz, whose name is on the screen, who is the developer of the first FDA clinical nano drug, which is Doxil. So the main questions that we are asking, and I will talk more about the methodology in my next presentation on Wednesday morning. The main issue that we're dealing with is the question which of these molecules could be discovered in silico to be a candidate drug, and we're dealing with a very large combinatorial space, if it's a combinatorial library, or very large chemical space. As you know, there are nearly 30 million commercially available drugs. And the methods that we use for, the general methods that are used for in silico are listed. Some of them are listed here: pharmacophore and structure-based and ligand-based and classifications and others. So those are some of the methods that we use. And as I told you, I'll talk about the algorithm that we developed much more on Wednesday. The main issue is to rationalize why is one set of molecules bioactive while another is not. So if that's a small group, there's a small group of bioactive in each disease, in each issue, in each biochemical test, there's a small group of molecules that is active, and there's a very large number of molecules that are not active, and we're trying to distinguish between those and those based only on the properties, on the molecular properties or physical chemical properties of one group versus the other. So learning and predicting in silico in general is starting with a set of molecules for learning, molecules for which we know their molecular activity or their biological activity. We transform those molecules into a set of properties which can be presented by numbers. We associate those numbers of properties with the numbers of the molecular activity, and then we have a model. And once we have a model, we can predict the activity of molecules with unknown activities, and this is the basic flow of what we're doing. So we're dealing with classification, and classification is the basis for distinguishing between actives and others which are not actives. But the more difficult task is to distinguish between more actives and less actives when molecules are quite similar. All of them are actives, but some are more active than the others. I am afraid that I replaced my my talk with the talk of Wednesday. I mixed between the two. And now I'll try to switch to the other one. Okay, then I'll continue today with the talk, with the general talk, and the liposomal talk will be on Wednesday. I'm sorry for this mistake. They're quite similar in the beginning, and then I found now that we're dealing with a more general talk now. So I've presented the learning and predicting in Silico and the issue of classification, and this is a general presentation of the algorithm, and this will be the most, probably the most detailed algorithmic part that I'll present. Our method is called Iterative Stochastic Elimination. It is a generic algorithm for solving extremely complex combinatorial problems. It is used for many protein. It was used for structure determination of proteins. It was used for docking molecules into proteins. And it was implemented recently for molecular discovery. It won the American Chemical Society Prize in 2000 for an emerging computational technology. And what we do is that we start with a large set of properties of the actives and the inactive molecules. We use random sampling of the properties in order to construct filters. So out of those 180 properties, we pick randomly 5 properties each time. We test their ability to distinguish between actives and inactives, and we throw out those in each iteration of hundreds of thousands samplings. We summarize this by checking which of all those properties is not contributing to the understanding of the difference between actives and inactives. And once we've done that, we reduce the number of properties, and we do this in iterations until we get to the point where we can compute all the remaining properties and check all of them, and then we get a set of solutions. We get an ensemble of solutions which can be used in order to predict molecular activities. So the best solutions ensemble is our model, and once we have that model, we can use it, and the model is a large set of filters, each which has a few property ranges. And now we pass a molecule, and a molecule that passes well through the filters, and those are the filters. When it passes more filters, it would get a positive number. When it does not pass well, it will get a negative number. And we are interested, of course, in the best molecules, and this is how the whole process works. So this is a summary of the whole process. We start with chemical structures. We transform them into mathematical descriptors of the properties. We know also the activity. So we use this iterative stochastic elimination engine to produce a model. And then we pass millions of molecules through this model in order to find which ones are the hits. Most of them would be rejected, and some would remain for as the top predicted actives. And those are some of the projects that we finished recently and have already found active molecules, because the molecules that we find at the top are not remaining in the computer, but are purchased or synthesized, mostly purchased and sent to laboratories around, to collaborating laboratories in order to test them for their activities. So one of our top results is finding, together with the Fraunhofer Institute, it's a collaboration between us and Fraunhofer Institute in Stuttgart, where we found toll-like receptor antagonists by screening about 2 million molecules through the models. We picked 60 out of those 2 million, and 90% of them, 56 out of the 60, were found to be antagonists. 5 of them are nanomolar antagonists, and 13 others are under 10 micromolar IC50s, and they're all patented now by a collaboration between Fraunhofer Institute and the Hebrew University. So here we had 90% success in predicting antagonists of the toll-like receptors. Another collaboration with a group in Frankfurt University, where we found peptides of 64 amino acids, which were designed in order to block the BCR-ABL in chronic myeloid leukemia cells. 6 out of 10 peptides that we synthesized were found to be active. In another project which was performed together with a few groups in an EU project called Cancer Grid, where there were 10 groups, 5 academics and 5 companies, we predicted differential cytotoxicity of compounds versus colon cancer cells versus human normal cells. And in this case, we sent 250 molecules for testing out of about 1 million. This was a low result compared to most of our others. So about 9% were found to be highly differential cytotoxicity versus the colon cancer versus the human cells. We have other results on multi-targeting. I will talk at length about the liposomal drug delivery system, where we have a very high validation, a success of our validations of the predictions of which molecules would load and be stable inside the liposomal drug delivery system, which is the basis also for the same nanoliposomes which are used for Doxil. And finally, collaboration with Novartis on PPAR delta, which is now being written up and published soon. So the nanoliposomal delivery system, which I'll talk about much more on Wednesday, we know that they are biocompatible, they are non-toxic. There are more than 10 liposomal drugs that have been approved by the FDA, and there are more than 20 that are now in advanced clinical studies. They benefit from the enhanced permeability and retention effect, which is typical of most cancer and many infections. So inflamed tissues and antibiotics could also benefit from this EPR effect. And the importance for changing the pharmacokinetics profile of the drugs is well known and appreciated. And a successful nanoliposomal drug should have a high drug concentration in nanoliposomes, which is expressed by the drug-to-lipid ratio. It needs to have a storage stability, and it has to release the drug in vivo in a pace that is appropriate for dealing with a disease condition. So this is the method that we load our nanoliposomes. It's what we call remote loading. So the remote loading is based on a gradient of a salt that is inside the liposomes. The drug enters whether it's a weak base. The drugs must be, in order to be remotely loaded, they have to be either weak bases or weak acids. And once the neutral drug, which is together with a protonated or the deprotonated part, passes the membrane, it reacts with the salt inside, and finally it's creates a solid solid sediment inside the liposome, and and that's how you can reach very high concentrations inside the lipid, and thus reach with a small amount of injection volume, you can reach a therapeutic dose. The modeling of liposome drug remote loading and loading stability was based on data sets of previous experiments. I won't go into all the details, and we used a few computational methods in collaboration with the University of North Carolina. This is a five-fold validation, external validation of our model. So there were, out of the negative ones, those that are not loaded properly, we only missed 5, and out of the positives, we missed 6 of them. And overall, it's a 92% were correctly classified, and we tested them also in our labs. We tested some of the positives, 2 of them, pravastatin and mupirocin, and then piroxicam, which is predicted to be negative, and those were all confirmed by us in the lab. And there were 10 others that were confirmed, except for one of them. They were confirmed by literature experiments. The same goes for the stability, although we had a much smaller set for testing the stability, and so the classification validation is a little bit less exact than the loading. But overall, it allowed us to do virtual screening of the whole comprehensive medicinal chemistry database of more than 8,500 molecules. And so this is the loading index on that scale, and the release index from our models on that scale. And we pick those that have the best loading and stability, and those are the most highly ranked. About 70 of those of that large database, about 70 molecules, less than 1%, have a high chance of being loaded and released from liposomes. And as this model was validated before, we are now looking into those 70s and looking into the different pharmacological activities. There's vasodilators and anti-migraine, antipsychotics. Not all of them, of course, would enjoy from being loaded into liposomes, but some of them will. We did the same for the MDL drug data report, and got from that, we also got about less than 1% of them were found to have high chances for being loaded and being stable in our nanoliposomes. The most interesting drug at this point of time, we found that mupirocin could be tested as a nano drug. And since mupirocin was until now limited to topical administration in the form of Bactroban, which may be known to many of you, it is rapidly eliminated and has high protein binding in systemic administration. So it is consumed very quickly. This is the red line for free mupirocin, while the nanomupirocin has a much longer profile in the pharmacokinetic profile, and the area under the curve is about 100 times better for the nanomupirocin compared to the free drug. And we also showed that in mice, it is effective in protecting mice against the challenge of bacterial necrotizing fasciitis model. And on the left side, let's see if we can activate this. So those are the untreated animals. On the anyway, for some reason, the left side is the untreated mice. They don't move. They stick together, and they are after their survival is quite short after the bacterial challenge, while those that got the protection of nanomupirocin remained stable. They were not affected by the bacterial challenge. So, in summary, our ISC predictions were validated by both statistical criteria and by experiments. The virtual screening process resulted in identifying about 1% of currently known drugs and those that are in advanced clinical studies from various pharmacological groups for this delivery system. And nanomupirocin is now patented for systemic delivery and is tested in a few labs around the world for its effects on endocarditis, on gonorrhea, and osteomyelitis. And our computational approach, which is generic, may guide the selection or the designing of good candidates in many different systems, including optimization of nanomaterials for specific tasks. I'd like to mention two companies that the Hebrew University constructed on the basis of our algorithm, and Pepticom is especially very active in designing peptides on the basis of known protein structures. Our supporters and collaborators in various projects, Fraunhofer in the Toll-like receptor, the Deutsche Forschungsgesellschaft in the BCR-ABL project, TargetX from Budapest in the cancer grid selectivity, and the chief scientist in the in the Ministry of Industry for the nanomupirocin. And I'd like to thank my collaborators, Professor Barnholtz, the inventor of Doxil,